So today I'm going to talk about the aniMotum package. This is a package that has been led and developed by Ian Johnson, and I've been lucky enough to be involved in some of the development of the package and assisted with the paper that will have been shared on the channels. And essentially, aniMotum is a package for quality control, behavioral estimation, and simulation of animal movement data. I came to it by getting stuck analyzing some seal movement data, which we'll talk about later on in this webinar. So, as a kind of outline of essentially what we're going to be going through, I'm going to do just a few slides just to kind of open the topic, discuss animal movement and different types of biologging data and the errors associated with those data, then introduce the aniMotum package quite briefly and essentially go straight into a practical where we can try our hands at working through some movement data within the aniMotum framework. So um, I imagine many of you on the call because it's an animal movement call, but me in particular, I've been fascinated by animal movement for a very long time, and I'm not the only one. Aristotle thought birds transformed from one species to another because the idea of migration hadn't really come about. And Homer thought that cranes traveled across the sky and rather than migrating, that they were on their way to battle pygmies. And I put up this picture because at the moment in Fife, in Scotland, where I'm based, we have large numbers of wintering geese. And I've been hearing them today because they're starting to get ready to migrate back north because they spend the winters here in Scotland, but they actually migrate back up to the Arctic Circle to breed. And understanding movements, we've come a long way, and we're now in a golden age of animal movement. There are large numbers of devices available, and there's a huge range of expertise within the scientific community for both deploying these tags and also understanding the data that they can collect. Animal movement data and biologging technology in particular have become a vital tool to understand both movement, behavior, and social interactions in foraging ecology. The first tags, I've said we've come a long way. The first tags were deployed back in the 60s. Radio transmitters and time depth recorders were deployed on animals. So that's 60 years ago. And the first satellite transmitters, the first PTTs, were deployed back in the 1990s. One of the first studies of animal movement that I can think of or can find is some of the first ringing studies, and they only happened at the start of the 1900s, and that was to understand the migration of swallows. So in terms of some fields, it's relatively new. There are now a broad range of methodological approaches to understand animal movement data. And the issue, certainly from my perspective, is that some of these technologies and some of these techniques to analyze the data are quite complicated, statistically speaking. And so for a non-specialist user, and during my PhD, when I first had my first biologging data set that I wanted to analyze, I really didn't know where to start. And I was a bit bamboozled by the range of different technologies or range of different techniques available to analyze that data. And so it can require quite a lot of investment from non-specialist users to try and analyze and process the data that a lot of these devices are able of collecting and transmitting. So I just want to talk about three kind of tracking technologies. They're three really common technologies, and they're three sets of data that can quite easily be moved into aniMotum. That's light-based geolocation, satellite transmitters, or PTTs, platform terminal transmitters, using the Argos network, and then GPS devices. So the first one is light-based geolocation. And I think the Greek explorer Pytheas was the first to measure latitude. He explored Europe in 325 BC and took measurements of the angle of the sun. So we know that if we can measure the angle of the sun, we can understand our latitude, and that's been done for quite a long time. But really, there was no real way of measuring longitude. And so you'd have to do it by dead reckoning. And so for some of you that might have done VHF radio transmitters, you'd understand dead reckoning or devices with magnetometers. If you know your bearing, you know your speed, and you know how long you've been traveling, you can use dead reckoning to understand your position. And that's what naval commanders did in the 1700s until 2 or 3 thousand men were lost at sea in the naval Scilly disaster. And that was essentially because they'd had bad weather for several days. They couldn't estimate their latitude. They didn't know what their longitude was, and they crashed into the Isles of Scilly off the southwest coast of England. But we know that the Earth is a globe, and there are 360 degrees in a sphere, and it takes us 24 hours for that sphere to rotate. So a 1 hour change or a 1 hour period of the day is equivalent to 15 degrees of rotation. So if you can build a good clock, and these are John Harrison's C clocks, but if you have an accurate clock that can maintain time over a long period of time, then that clock you'll have set to Greenwich meridian, to Greenwich mean time, and then you'll have a second clock that you adjust to local midday. And the difference in time between those two is your difference in longitude. Now, of course, we don't stick massive clocks to the back of animals. We use tiny geolocation devices. So in several hundred years of technological development, we've now got teeny tiny sensors that we can put on these animals, and they'll record just the light level and a clock to measure time. So these devices, using the ideas behind geolocation that I've just mentioned, are accurate typically to within 200 kilometers or less than 1 degree. They're not highly accurate, but they last a very long time and they're very small, so you can put them on a wide range of animals. But, and this is critical for our understanding of error in the positions of animals and why we might need to use aniMotum, they're vulnerable to things like shading. So if the animal tucks its leg under a wing, or if the animal sat on the water at sunrise and sunset, then we're going to get a really bad estimate of the timing of sunrise and sunset. For example, where I am in Scotland, there's only a 4-minute difference between sunset here, which has basically just happened, and sunset on the west coast of Scotland. They're also vulnerable to anthropogenic light. So if you've got birds that hang around fishing vessels at sea or birds that hang around oil rigs, you're going to get strange light at night, which will also interfere with your understanding of position. The second set of data that we could use in aniMotum is tracking using radio waves, and that's using platform terminal transmitters. So the Argos satellite system, the first satellites were put up in the late 70s, and these estimate your location from Doppler shift. So if you imagine you're walking down the road and an ambulance goes past, you'll hear that change in pitch as the ambulance passes you. It goes from a high pitch to a low pitch. And that's exactly what happens with a satellite transmitter. So it's transmitting a radio wave, your satellite goes overhead, and the transmitter and the satellite are communicating with one another, and the satellite's listening out for a change in the shift in that transmission. Now, the more locations we have, or the more messages that are passed between the satellite and the transmitter over the course of the 10 or 15 minutes that the satellite's in view, the more accurate we are in the location. And for those of you that have worked with satellite transmitters, you'll know that you'll get a location class, which is typically like a 321 or I think ABZ. Those are the kind of classes that you get. And they essentially represent the number of locations that you've got. Now, in my case, I work on marine animals who may not spend 10 or 15 minutes at the surface. And there's no reason to say that they'll be at the surface at the same time that the satellite goes overhead. And this was one of the reasons that I actually first got in contact with Ian and started talking to him about what became aniMotum. We would put satellite transmitters on seals, and I'd get large gaps in my transmissions. And what I didn't know until I saw this tweet was that harp seals spend an awful lot of their time swimming upside down. And so our transmitters that we put on the back of their heads actually weren't seeing the sky very often. So frustrating, kind of interesting at the same time. Then finally, we've got GPS. This is clocks again, a lot like our longitude problem, but it's atomic clocks. So there's a constellation of GPS satellites that you'll all be familiar with because your mobile phones are communicating with them quite a lot. And they are all transmitting their position in the constellation in space and the current time of their atomic clock. And then your GPS receiver, and here's a couple of pictures of GPS receivers that I've used to track the movements of gannets, northern gannets in the UK. The antenna on that, the receiver, will triangulate its position based on detecting signals from typically a minimum of 4 satellites. Now, doing that is very battery intensive. it's only a few hundred kilometers to the Argos satellites, but it's 20,000 kilometers to the GPS satellites. So your tags tend not to last very long. You can have problems where they miss fixes. And obviously they need to see the sky. And so they're vulnerable to things like canopy shading, or if, again, similar to our PTTs and our seals, they're vulnerable if your animal dives and spends time under the water. So all of these technologies are a fantastic way of understanding and tracking animals around our globe and understanding their movements, understanding their behavior. But crucially, they've all got some source of error, or they've got some sort of issues that mean that you may not get regular good locations that you can use. So if we want to analyze our animal movement data, our telemetry data, we can use the state-space model paradigm. And we're going to think about our animal movements inside the paradigm of state-space models. Now, as I said at the start, I'm not really a statistician. There have been some very, very good talks within this webinar series already on state-space models, including by Vianney and by Teo Michelot. And there's been some great talks by Maria Gomete on TMB, Template Model Builder. And so I would suggest if you're interested in some of the things that I'm talking about, you might want to go back and have a look at some of those seminars as well. But essentially, although we know the exact location, I remember where I was when I put that tag on the back of that seal. Once we leave that seal on the ice, once we leave our study animal at the breeding colony, or we release it after we've captured it, we very rarely see it. I've spent quite a lot of time sat on seabird islands with a pair of binoculars trying to find one of our tagged birds. But when you've got 100,000 birds and you've tagged 10 or 20 of them, it's very unlikely that you're ever going to see them. So what we can think, or how we can frame this, is that our recorded locations or our transmitted locations are essentially an observation of a hidden state, and that hidden state is the true location of the animal. And that true location we've observed with some error associated with it. And so that's the error that I've just described from those three previous techniques. So our state-space model, when we start to think about what we're doing with aniMotum, has two parts. The first part is a movement process model. So this could be a random walk or a correlated random walk. In the case of aniMotum, we're fitting these in continuous time. So it would be a continuous time correlated random walk. And then our second part is a measurement model. And that measurement model has information about the error associated with the location measurement that we've taken. And what we're doing with this state-space model is we're trying to separate the variation in movement from variation in the measurement of that movement. And I think for me, a particular light bulb moment was when I was reading the paper in Tree that Toby Patterson and colleagues published in 2008. So if you look at this figure and you look just first at the black crosses, you can think of that as being what we're going to be getting back from our satellite transmitters. We're getting back noisy locations that aren't the true location of the animal, but we have an understanding, at least, hopefully, of the error in that location. Now, with some information about how the animal might move in terms of a correlated random walk or a random walk or something else, and some information about the error associated with the measurement of that location, we can condition a model, train a model, depending on the language you want to use, to try and recover the true path. So in the paper, they kind of demonstrate this and show that they can recover the blue line, and the blue shading then is error around their estimated path of the animal. And then they can compare it with the red path, which is the truth. So they do quite a good job with their model of getting somewhere close to the true path of that animal using a state-space model, and some information on the type of movement and some information on the error associated with the measurement. For those of you that might be interested in equations, then this is kind of a way of thinking about, algebraically thinking about our aniMotum model, or one of the models within aniMotum, I should say. So we have our continuous time movement process model that will be a random walk or a correlated random walk. And essentially, we're estimating the true location as a function of the previous true location plus a measurement of the velocity along the path, because we're going to start looking at correlations in velocity. The relative time from the last location, as I say, this is in continuous time, so that time step can vary between locations, and then some randomness in the movement. And then our second model in our state space model is our observation model, where we include information about the error. So that could be information about the geolocation error, or information about the least squares, those 3 2s and 1s for the Argos locations, or more recently the Kalman filter for those of you that might have been processing satellite transmitter data more recently. Or it can be a mixture of both. And we can also fit GPS data within these, assuming small errors around the GPS locations. So, aniMotum. I would really encourage you to look at the documentation around the package. This is going to be quite a whistle-stop tour, if you like, of what aniMotum can do. Just a first pass at some of its abilities. We had a paper published a couple of years ago now in Methods in Ecology and Evolution that does a really good job of describing and walking you through the package, sorry. And then there are an awful lot of supplementary materials associated with it, with some guides on how to analyze data, how to replicate the analyses that we do in that paper, as well as then all the vignettes and guidance that are associated with the package and on Ian's GitHub page. But essentially, our workflow is to bring into R some data that we want to try and fit our state space model to. This can be Argos least squares data, Argos Kalman filter data, GPS data, or geolocation data. I'd say now that it really works best for above air geolocation, if you like. So the kind of geolocators that you might have used to track bird migration. This isn't really optimized for working on those of you that might be tracking fish using geolocation. So there are issues with the errors associated with those geolocation positions, because the error in the light, if you think about it that way, isn't just error in latitude and longitude, it's also how light attenuates with depth. So we'll load in our data, we'll formulate our data, we'll format our data to make sure that it's in the right format. We'll pass what kind of location class we have for our Kalman filters, or whether or not it's geolocation data or GPS data. Then we will choose our movement model to fit our state space model to. So this could be a random walk, a correlated random walk, or more recently, Ian has fitted a move persistence model that allows us to estimate changes in behavior at the same time as estimating the path of the animal. Then we'll check our model fit, try and make sure that our model's fitting quite well, look at histograms of look at plots of our residuals. Those are residuals calculated using one step ahead residuals. And then we can do things like visualize our models and map the outputs, pull out the regularized locations. And then finally, although I don't know that I'm going to get time to go onto it today, we can simulate from those models. So for those of you that might be interested in generating background points for some kind of habitat model, or generating simulations to make sure that your model's fitting the data well, there are a range of different ways of simulating from the fitted model. And there was a great package released recently by Josh London that looked to reroute movement paths around land masses. So a lot of my work in the past has been marine, and so we have issues where animals need to swim around a landmass or around a peninsula, and some of these models will often ignore that. And so the path rerouter package, we've incorporated some of its functions into aniMotum, and so allowing us to reroute our movement paths or our simulated paths around land. So with that, I'll move on to the practical. So I'm going to assume that you've already downloaded aniMotum. If you haven't already had a look at the practical, you can go to the GitHub, go to my Grecian aniMotum folder, and then there's the aniMotum webinar tutorial as an HTML file. If you click through to view it in RAW, you'll need to download it from here to get the actual HTML version of the practical. I tried earlier, and if you download it any other way, you end up with a text file full of HTML script, which is slightly less pretty to work your way through. So, as I said, this tutorial is heavily based on the appendices supporting information of the aniMotum paper. So go through this practical. We'll go through it now, but obviously go through it again in your own time. If you get stuck, maybe check out the supplementary material first. There's also the GitHub package page. And so people that have got stuck in the past have gone there and posted questions, so you can double check whether or not the issue that you're running into is an issue that's also been faced by someone in the past, and likely there'll be an answer there for you. So the idea, as I said, was to load in an example data set, fit the state-space model, visualize the model fit, look at estimating move persistence, extract all our regularized data, which you might then want to pass on to a species distribution model, or you might want to pass on to doing some mapping yourself. You might have a particular style of mapping that is different from the native mapping that you can do, the functionality that the package already has. And then there probably won't be time for doing simulations and rerouting of paths, but we'll see how we get on.
James, would you mind making your text font a little bit bigger? Great, thanks.
You mean like I had before and then have lost it?
Yes.
So I'm hoping that you're familiar with using the tidyverse style of coding. That's how I code. And I use pipes to do data transformations. And we'll be using the SF package for spatial handling, spatial aspects of the project. aniMotum now deals with SF objects quite nicely, so you can move between SF and aniMotum and tidyverse really nicely. If you're not too sure about tidyverse and you're currently sat there saying tidy what, then the package and the book are really useful, really nice way of thinking about data science. Personally, I think a really good way of learning to code in R, and gives you a lot of transferable skills moving forward with your careers. And as I said, there's a link there for the aniMotum package. So back in March 2019, I was out in the Gulf of St. Lawrence, and I was tracking seals as part of a research project that was led from collaborators here in the UK. And we put a number of satellite transmitters on juvenile harp seals to try and understand their first migrations up into the Arctic. I published the paper that resulted from that work a couple of years ago now, in Royal Society Open Science. So it's all open access. The data and the code are also all available there as well. So again, it's another resource for those of you that might be interested in using aniMotum for your own analyses. For those of you that might be interested in what it's like going out and tracking harp seals, we were actually filmed by the BBC as they were part of Frozen Planet 2, which was a landmark BBC documentary series that aired here last year. And I think in the US, the end of last year, it aired. I think you can catch up on various streaming platforms. But yeah, there's some great footage of harp seals in one of the episodes. And then the final episode, there's also some interviews with us about the work that we were doing. So great resource. So I would load aniMotum into your workspace along with SF and tidyverse, as we've just said. I've also loaded in the patchwork package. That's just a nice way of stitching together plots. Just makes it a bit easy to compare things. So in the repository, I've left a CSV file called harps with the raw data from 3 of the tags that we put out back in 2019. So if you load that in with read CSV and then just type in locks into the terminal, it should spit out the top 10 rows of the tibble. You'll see that there's 11,000 best part of locations. and they're in a relatively standard format. So aniMotum is looking for data in this format with a column for ID, a column for date, and a column for location class. Yeah, so kind of a standard format for this kind of data is you'll have a column for ID, a column for date time, a column for the location classes. So these are the threes, twos, ones, Bs that I previously talked about. So these are the least squares, Argos error. And then you've got your longitude and your latitude estimates. For more recent satellite transmitter data, you'll have Kalman filtered locations. And so then you've got the error around the semi-major and semi-minor, so the error ellipses. So these are the measurements of the error of the ellipse. And then I'm not quite sure off the top of my head which one that one is. The Argos error ellipse orientation. There you go. So your data should look like that. If you're working with geolocated data or GPS data, then aniMotum still expects this column, these first columns, location class, if you look at the documentation, would be changed with like a GL or a G, depending on geolocation or GPS. And then the long and lat columns, and then these would be replaced with the error in your geolocation estimate, for example, if you have one. So that's the details there. There's a paper that we published back in 2020 that discusses how we came up with the way of dealing with the error. So we did some double tagging studies with GPS and satellite transmitter data that helps refine the model. So then let's load this in and have a quick plot and have a look. And you can see that we have one track with some big gaps. So this is one of our seals that most likely swam. Well, I know pretty sure this gap here, might be that gap there, is it crossing over from Newfoundland over to Greenland. And so when they travel in for extended periods in a particular direction, they often swim upside down. So this is probably what's going on here. And then we've got these two tracks, and you can see at least one location on each of those plots that doesn't look right. So we can pass these as they pretty much are to aniMotum and use a state space model to deal with some of these errors, to regularize the data to, say, daily locations. But the question typically would be, well, how frequently should we ask for our regularized data? And so one way to do that is just this line of code here. So we just group our data by the individual ID, and then we ask it to summarize the difference in time between each successive location. So each successive row. So what's the difference in time between row 1 and row n and row n minus 1? Calculate the mean, convert it to hours, because otherwise it'll be in seconds, and then print that out and then calculate the maximum. So what we get from that line of code is the mean interval between our locations from the transmitted data in that data frame. So the first animal, on average, it's a PTT location every 3 hours. The second animal, it's every 1 hour. And the third animal, it's a little over every 1 hour. But if you look at the maximum interval that we've got for that first animal, the biggest difference in time between successive locations is 303 hours. So that's 12 and a half days. And so that's probably these big gaps just here. So in the paper, I trimmed the data and got rid of some of these big gaps because I was interested in combining it with the dive data that the tags were also transmitting. So I wanted it 6-hour locations because we have 6-hour dive summaries. But in this case, we'll put that to one side for speed and simplicity, and we'll just ask the model for daily locations. And we'll have to accept that we're going to be interpolating over kind of 12 days in one extreme case. So you just pass that data frame to aniMotum. We use the fit SSM because we want to fit a state space model. We can specify a Vmax, so the maximum likely travel speed that's used in a pre-filtering step, and that's in meters a second. So in this case, we're saying that any locations that suggest the animals had to travel faster than 5 meters a second, which is quite quick in water, we can get rid of. And then we're going to fit a correlated random walk. So this is a continuous time correlated random walk, 24 hours. And then this is simply a line of code that stops aniMotum from spitting out all the iterations as it's running through. So otherwise the console would just be filled with output. And this is the kind of output that we get. So one thing I wanted to kind of highlight by doing this was, and I'll just run this so that you can see, was when I first started looking at state-space models, we were fitting them in a Bayesian framework in MCMC, and it would take us hours, maybe even days, to fit a path to one or two animals. Now it runs in the time it takes me to complain about how bad it used to be. So we have a model converged for 3. So I was fitting this recently to 80 animals, and it takes time, but it takes 20 or 30 minutes on my relatively new MacBook Pro compared to weeks if you were trying to fit this using Markov chain Monte Carlo approaches. So we can ask for a summary of our model output, and it will tell us some characteristics of the model. So we get an AIC, how well the model's fitting, or some measure of that, the kind of thing that we're looking for, what's been filtered, etc., 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 and some parameter estimates for the correlated random walk that we fitted in this case. aniMotum comes with a whole load of really handy plotting functions that Ian's developed. So first off, we can ask just for a simple plot of the model. We can ask to look at the predicted locations. So in our case, that'll be our 24-hour locations, or we can ask for the fitted locations, which would be the ones that the model has used. Excuse me. So those would be the irregular locations. So what you can see here is a plot of the predicted locations in X and Y space separately. And for this first animal, where we know that we were going to try and predict over 12-day gaps, you can see these big orange ellipses. So essentially the model is showing us, yep, sure, I can essentially interpolate over the 2 weeks for you, but I've got quite a lot of uncertainty because in the best part of 2 weeks, the animal could travel quite a lot of different ways to get between A and B. We can then plot, so this is just changing type 1, which is the X and Y plot separately, to type 2, which is the X and Y plots as essentially as a map. And what you're then seeing here is those errors in 2D space rather than in 1D space. And so you can see for this first animal, the model is really doing a good job of showing us how much uncertainty there is in trying to linearly interpolate across these gaps, which is why when it came to the paper, I essentially dropped a lot of the data from this animal. But we seem to have a much better fit where we've got more regular locations, and you can see the regular 24-hour orange dots on this middle plot. If you want to map it, then you can just there's a map function within aniMotum, and that will use an inbuilt shapefile, global shapefile, and plot each of the tracks. And again, you can ask for the fitted locations, which would be irregularly spaced based on the data that you put into the model, or you can ask for the predicted locations, which will be the sampled at the time step that you've asked the model for. So in our case, these are the 24-hour locations. Now, often we want to try and understand how well these models fit. And one approach is to just do these kind of plots and look for gaps and look for errors and issues. Another one is to look at residual plots. So I would suggest you don't try and run this code within this session. What the model is doing to assess residual fit is essentially predicting one step ahead for each of the data points for each of the animals that you have, and then assessing how well it's doing at predicting one step ahead. So these are one step ahead residuals. There's some more details in the paper that would do a better job of explaining it than me, but it's a really useful way of assessing the performance of the model. And so we just use the OSAR, one step ahead residual function, which in my case took about 20 minutes to run on these three animals. And then we can plot a time series of the residuals. So again, in X and Y space, we can plot QQ plots, or we can plot an autocorrelation function. So if you have a look in the paper, Ian explains how you could fit a random walk and you might still see some patterning in an autocorrelation plot of the residuals. But when you then fit a correlated random walk, that goes away. So it's just it's a really nice way of comparing model fits between the different models that we have within aniMotum. This plot in the middle suggests that the correlated random walk's not doing a great job necessarily at predicting at very small time steps and very long time steps. That's why you've got these kind of big tails. So the next thing that you might want to do, one of the other things that is kind of the key things that we can do within aniMotum, is to fit move persistence models. Now, this is a way of inferring changes in behavior along the movement path. The key thing to say is it's a continuous behavioral index, and it's essentially looking at changes in the autocorrelation in velocity. So autocorrelation in both speed and direction along a path. So if you look at your track, you might have periods where the animal looks like it's traveling quite fast and quite directed, and you might have periods of a path where an animal looks like it's traveling quite slow, and maybe the turning angles between successive locations are quite high. It looks more like a random walk than a correlated random walk. And so you get a measure out between 0 and 1 that kind of measures the changes within that space, if you like. There's a great paper in Ecology 2019 where Ian does again a better job of describing at least of describing in more detail how this works. I've said it's a continuous behavior index because it's quite different from hidden Markov models as a way of analyzing animal movement data and trying to look at discrete states. So again, as I mentioned in the talk, you might want to refer to previous talks, previous webinars within this series that have talked about using hidden Markov models for animal movement. So there are two ways of fitting move persistence. You can either use the function fit npm for fitting a move persistence model, which you would fit to the results that you get out of your state space model. Or more recently, you can fit the move persistence instead of fitting the correlated random walk or the random walk in the original call to the state space model. The difference being there that you are estimating move persistence at the same time as estimating the position of the animal. So the move persistence measure incorporates some of the uncertainty that you have in the animal's location. If you use the fit npm function, then you have the option of estimating move persistence individually for each animal, or by doing a joint move persistence model where you have a shared parameter that allows you to kind of maybe understand something about all of the animals as a group. And so, as we say, we try to keep the way that you run these models relatively straightforward and relatively familiar. So you pass, in this case, I would pass the fit object that's come out of the state space model for the seal data, pass it to fit npm, and tell it to fit it to the predicted. So those are the regular locations. And in this case, I've said the joint move persistence model. And so again, that should fit quite quickly. I'll not do the control change at the end so that you can see how quickly it fits. Oh, brilliant. So these are the kind of outputs that you would expect to get from the call to fit npm. So these are our plots, our time series plots of move persistence. So what you can see here is periods of residency and periods of transience. So we've got times when the animal's moving quite directed between potentially between foraging patches or between migratory stopover spots. And then we've got periods where the animal seems to be more sedentary, moving slower, moving less directed. You see how this changes over time. It's also possible to link mood persistence to the environment, but that's beyond the scope of this talk. There's a paper by Ian that you can have a look at, or if you go to the Harpsfield paper that I referenced at the start of the talk, the Royal Society Open Science paper, then I do that as well. So then we're trying to understand relationships between the environment and mood persistence. So in the case of the harp seals, are there particular environmental characteristics that drive changes in mood persistence? Then data extraction is great, being able to fit these models, but what do we do with the data that comes out of them? Well, there's a great function called grab that will pull out the predicted or the fitted locations for you. So you can easily create a data frame that you can pass on to other analyses. So in the case of the state space model, you could pull out the 24-hour regularized locations by saying grab predicted. And in the case of the move persistence model, you could say grab fitted, which are the move persistence estimates for each of those that have been fitted to each of those regularized locations. And then using the left join tidyverse command, I'm just combining those two data frames one to the other, and you'll get a tibble that is the ID of the animals in question, the regularized date time, longitude, latitude, and X and Y, which are our regularized positions, the error associated with those positions, and then you can't see it because of the width of the plot, but G, so gamma, which is our measure of move persistence, and a measure of error in gamma as well. Now, for me, I want to do this because I have a particular way of that I like to do my maps and my plots in R. So I would pull those locations out and then I would pass them to ggplot to create a map. And actually, a lot of this code is basically what's underlying the map function within aniMotum, because this code be passed on and became part of the package. But this is just so that you can see what's going on and how you might want to do it yourself or tweak plots. So I'm using the R Natural Earth package, which is a great resource for shapefiles of the globe. I'm just going to pull in a OK scaled shapefile, and then I'm going to define a projection. In this case, obviously this webinar isn't about mapping, so I'm not going to go into too much detail, but essentially this is a Lambeth azimuthal equal area projection that I've centered on the center of the study area, so 60 degrees north and 50 degrees west. I pull out the longitude and latitude coordinates and tell SF that that's what they are. And so I can create a SF object from my data frame, and I can set the coordinate system to WGS 1984, so just standard latitude and longitudes, and just using that code. Again, there are some reasons why you might want to do that that we may not get into, but essentially SF likes you using codes rather than defining long character strings. And then we can set up a ggplot, which for those of you that use ggplot is relatively straightforward. So we have a call to ggplot. In this case, I've set a black and white theme. We pass the global shapefile. So this is our shapefile world, which is an SF object, but is in long and lat. ggplot's going to do an on-the-fly transformation for us. We pass our spatial object, our SF object, which is our regularized locations, and we ask it to color those locations by our gamma estimate. So again, we're passing this to SF, so it's going to do all the transformations on the fly for us. I'm using the Viridis color palette because it's fashionable and does a very good job of distinguishing between light and dark across a range of different visual people with different visual preferences. Then I will specify a box that I want to trim my plot to. So in this case, these are the X, Y, UTM coordinates of a box that zeroes in on the study site, and here's the projection that we're using. So then ggplot and SF on the fly are doing all the transformations for me to make it into a nice Lambert azimuthal equal area projection. And what I've just done here is faceted it by ID. So you can pull out those 3 different plots, one for each animal in a nice grid. So What this then allows us to have a look at is we've started by looking at our tracks. We've then regularized our tracks and we've plotted the regularized locations. We've then run a move persistence model and we've looked at a time series plots of our move persistence. But by doing this, we can then look in 2D space at our move persistence and how it changes along the migration path. And so what I hope this will help you illustrate is that we've got periods of higher, more direct or higher speeds, more directed travel as the animals are crossing over the Labrador Sea. And then we've got periods of slower travel, less directed travel, periods of low persistence in their movement when they're either resting or foraging on their migration. So that's the kind of thing that I would do to visualize a move persistence model or a state space model output. So nice job. Good plot. Cute seal, because why not? With a little bit of time we've got left, I think, I just want to highlight an example of what you might do with GPS data. We won't do GPS and geolocation, we'll just do GPS. So in the repository, there's also, actually, it's not in the repository. So in the repository for the supplementary materials for the aniMotum paper, there is the GPS data. You can just load it straight into R using that that HTML link, that URL. That's what I was looking for. So these are little penguin GPS data. You can load them in with read CSV with that link, and you can just double check what they look like. So I talked a lot about how you want to make sure that the data that's going into aniMotum looks okay. So yeah, the key thing to note here is that when you're using either GPS data or when you're using geolocation data, that you want to make sure that your location class column is in the right format. So in this case, we're using GPS data. So that is just a vector of Gs, just to tell aniMotum that it's GPS data. So we can load that in, and exactly the same as before with the seal data, we specify a Vmax, a model, correlated random walk model. Here we've got a minimum time between locations that's been specified because you might have repeated locations. So quite often with GPS data, you'll get double fixes. So if you go through your data time series, you'll have more than one identical fix. So this is just thinning the data that goes into aniMotum. And then you'll remember that I said that aniMotum wants hourly locations, or how many hours you want between successive locations, which in the case of a satellite transmitter, when you might want daily locations, makes sense. But obviously with a little penguin with GPS data, we've got much more regular fixes. And so here I'm just saying I want 2-minute locations. So 2 divided by 60, 2-minute locations. And then we'll take the output of that and fit it to our move persistence model. And then here's a plot just to give you an idea of what the output might look like. So we've got our 2-minute predicted tracks for our little penguins, and we've got our regularized move persistence estimates. Again, that model should fit quite well, quite quickly. It might take a little bit longer to run than the harp seals because it's got a lot more data that it's dealing with. One thing just to highlight here is that aniMotum will sometimes struggle with GPS data to estimate one of the parameters in the uncertainty. If you get errors coming out, then you can just add this line of code into your call to fit SSM, and that should resolve that error. And then just some final thoughts, really. I'm guilty of doing this myself, but it's sometimes easy to forget that there's a lot of statistical heavy lifting that's going on in the background when you run these models. We try our best to make them really simple to use and really straightforward to use, but actually in the background it's doing a continuous time correlated random walk. It's fitting it all. Yeah, there's a lot going on. So if I think back to how I used to approach some of these analyses when I was doing things with MCMC or doing a Bayesian approach where it might take several days for my model to run, I'd be very careful with how my data were formatted going in. Whereas now it's quite easy just to throw your data into the model and see what happens. And so I just caution against that. You might get some slightly unusual results, or you'd get some unexplained issues that you might face. So just be really careful, go through the ideas that we've talked about today, make sure your data is formatted correctly. All the functions are really well documented, and it'll be highlighted within each of the functions how the data should be structured that's going into the model and what you can expect as an output. Yeah, as I've talked about with the location classes, and as talked about in my slides earlier, the geolocation data, Argos data, and GPS data have different errors associated with the locations, different uncertainties, and how they're dealt with within aniMotum is different. So you need to make sure that you're using the right error structure for the data that you're inputting. And aniMotum won't know what to do with it unless you specify the location class column correctly. Um, so yeah, I hope the practical has been useful. I hope it's given you an insight into how we can use aniMotum, how aniMotum can help you with your own data analyses. It's aimed to be simple and straightforward to use. I hope this has given you an indication that you can get quite far analyzing some of your data from a relatively low bar, but there are also some great things within there that are much more complicated that if you start getting into it and want to look in more detail, then you can explore different routes. Yeah, with that, thank you very much. There are some references at the bottom to the papers that we referred to, and a link to the BBC Earth website if you want to have a look at Frozen Planet 2. I don't know where you get your streams from, but I'm sure you'll be able to find it online somewhere.
All right. Thanks, James. That was great. We've got a number of questions in Poll Everywhere, and so I can just go through those and yeah, see what you can help us with. And I don't think I'm going to go straight through them from the bottom to the top, or top to the bottom, because there was one question. There's a couple of questions since you've mentioned you have the GPS example and the Argos example. There was a question is, can you run this with both the GPS and Argos in the same file? You're still muted.
Yeah, you should be able to, yes. As long as so the model fit will, well, no, sorry, I'll say that again. As the model is being fit, the model is looking at the location class column and is deciding how best to deal with that error. The model definitely runs with different Kalman filter. So if you've got Kalman filter and least squares for your PTTs, it will just flip between the two. And I'm pretty sure I've run it with GPS as well. So as long as in the location class for the GPS fix, you've got G, then aniMotum will know that that row in your data frame, that particular fix, has a much higher level of certainty in its position compared with the other locations.
Great. And I think related to that, there's a question about the G in the location class. Is aniMotum assuming similar error structures for fast lock positions and true GPS positions?
Yes. I think it treats all GPS positions as the same. That's something I can go to Ian and double check, but I don't think we distinguish between fast lock and standard GPS at the moment.
Okay, great.
I think you can add in your own error. So if you've got a fast lock location, you might be able to change how uncertain you are with the location with the X and Y standard errors.
Okay. Thinking about error, but in a different concept. There's been a couple of people that have gotten a couple of errors, I think, going through. So I'm putting in the chat one of them that's come up a couple of times. And I don't know if that's something that there was two of them there. If there's any, or if those are errors that need to be looked at outside of
That
this.
could be if someone that's run into that error, is that them running it on their own data, or is that them running it on either the
I
package data or the example data?
I
Because it
assume it's the example data.
so it could be an issue with the installation, whether or not the matrix package that's loaded is correct, and whether or not the version of TMB that's been loaded is correct. Um, there are some issues when you install aniMotum for the first time that you have all your ducks in a row, if you like, for the correct versions of all of those packages. You can download. Sorry, my dog is in the process of pulling my laptop off my desk. Um, you can load the binary version of the package by downloading it as a zip file off the GitHub page um, or the iUniverse page, and that might solve it for you. But it's common to run into a couple of issues when you first download aniMotum.
Okay, great. Thanks. Let's
Sorry,
see.
just to say it, just so there may be, I think there's a help file or a description of that problem on the GitHub page that you can double check against.
Great. There was a question about, can you expand on how the move persistence model is different from hidden Markov models?
Yeah, so in a hidden Markov model, this is with a caveat of this is as I understand it, and I am happy to be corrected. In a hidden Markov model, what you're essentially doing is you're conditioning, say, three different random walks. So in a three-state model, you'll have three different distributions of step lengths and turning angles. And at any time along your time series of locations, your model can decide that that location comes from one of those three different locations. It's kind of a backward way of thinking about it, but essentially you're trying to classify each of those locations into one of three states or one of five states or one of two states. What what we're doing with move persistence instead is to say that there's some kind of continuum of behavior where you're what you could call a transiting state. You're directed and traveling quickly, and then there's a residency state where you're moving slowly and less directed. But that you're on a continuum of behavior between those rather than there being a threshold to cut off between one or the other. Um, in my head, I sometimes think of it that if I have like high resolution GPS data where I'm really sure that I've captured a lot of that behavior, then a hidden Markov model is a really useful way of distinguishing between multiple different behaviors. Whereas if I've got daily locations and I'm interested in broad scale migratory movements, move persistence might be more appropriate just as some kind of continuum of behavior where they're more likely to be traveling or they're more likely to be resident.
Great. This question asks or states that they've seen different approaches to the Vmax filter in the SDA filtering process. Some use very conservative or high speeds, and others use fairly restrictive speeds. For example, limiting gray seal movement to 2 meters per second, which filters out a lot of Argos locations. And they're asking, what is your approach?
I would say that there isn't a right and wrong answer, and that's not meant to sound like a cop out. Um, Animals will move differently. Different seals within the same species will move differently. Um, a very conservative speed threshold will dump a lot of data. Um, that might be okay in your case because you've got lots. Essentially, I would say maybe start high, run some models, and then if you're still getting, so in the case of the harp seals, if I started very high, I'd get some of those strange outliers included in the model because there's enough time for the animals to get there. And so I'd then just start lowering that threshold of speed. And typically you'll get to a threshold where increasing it more doesn't really change the output that you're getting from your model. But I'm afraid it's not really black and white. It's more of a dial that you can tune.
Sure. I think there's a similar question, similar, probably not black and white, but the questions for dealing with data gaps, one might segment the track or trip into segments where the time difference between successive positions does not exceed, say, 12 hours, 24, 48, or even 10 days. How does one determine an appropriate data gap tolerance?
Yes. How does one? Um, I guess, I mean, on a practical point of view, it's how happy are you to be interpolating data? I mean, you don't really know where the animal's gone in those times. So you might say, well, if the gap's a couple of days and I'm interested in 24-hour locations, then I can interpolate through those because the animal can't get too far away. But if you've got long gaps of a couple of weeks, then the animal can do an awful lot. Um, there are developing methods to deal with this problem. So within the momentum package, one of the hidden Markov model packages, there's the option for multiple imputation. So you run multiple tracks, and then obviously each path of those imputations has a different possibility of how far away it can get between by crossing your data gap. And then I think what that package does, I think Teo may describe it in the previous webinar, is fit a hidden Markov model to each of those tracks to estimate behavior and then kind of averages across them, if you like. So there are approaches that might allow you to incorporate larger gaps, but I used to hate the phrase when people used to say it to me, but I guess it depends on the question. Like it depends what you're actually trying to do with your data. If you want to make a pretty map, then you can probably interpolate, but you'll end up with a very straight line and it's not really telling you anything. Um, aniMotum is quite happy for you to pass segmented tracks and deal with them individually. So you could just create sub tracks from that path. So you say 3 tracks from your one track, and then fit your model to each of those tracks in turn, and just acknowledge within the paper or within your study that you have gaps, and you feel the most honest way of dealing with it is to show those gaps.
Great. Uh, in terms of model diagnostics and model selection, when comparing the one step ahead residuals, is there a model assumption that is worse to violate? Should this person be prioritizing model fits that say look better for residual heteroscedasticity, normality, or autocorrelation?
Not that I'm aware of. I would try and take a more holistic look at those three as a guide. I don't think it's necessarily black and white. You want to think about the data that you have, think about what you're trying to do with your, say, your correlated random walk or your random walk. Those initial visual plots will give you an idea of how well the models will fit. One example would be when I had large gaps in some of my harp seal tracks, a correlated random walk ended up with very strange loopy artifacts. When I know the model's just not fitting very well, I don't need to do one step ahead residuals to know that the model's not fitting the data very well there. Um, I would look at those 3 plots. I'm just going to scroll back up to where it was. I'd look at them together, if you like. To be honest, there's a suggestion with this model that actually the skew here would suggest that maybe the model's not fitting the data as well towards the end of the migration than it is at the start. Um, so yeah, I would look at all 3 different plots. I'd have a think about your analysis, some of the initial plots that I showed, and maybe take those all as a broader picture. But I'm not sure there's a hard and fast black and white rule.
Do you have experience or do you know anyone else that's used aniMotum on acoustic telemetry data?
Ian, I think, not to throw him in it. I haven't personally, no. I'm not that familiar with acoustic telemetry data.
Okay. I think it sounds like after estimating positions using another R package, I think this person imagines it wouldn't be much different from GPS. Okay, but maybe Ian's the
So
one.
yeah, I mean, so with the if you were to fit a aniMotum model to GPS data, I'm pretty sure that you can add your own measures of error. And so in those cases, if you have some error from the previous model that you could pass on, then you could use those as some measure of standard error around the location estimates.
What's the difference between Argos and Argos LS data?
So there are two types of, so all the data will be Argos data. If that's my mistake in the language that I've been using, then I'm sorry. There are two types of error that get reported by Argos. One is least squares, which is the 321 zeros, ABZs. And then the new one is the Kalman filtered data that goes through another level of processing. And then that gives you these error ellipses. So the two types of Argos data that you might get from CLS would be lats and longs with either the Argos least squares error or the Argos Kalman filtered error.
And can you get both of those at the same time, or you have to choose one or the other?
You'll always get uh, you'll always get least squares, which is like the older version, the 321s. I think all modern tags now get the Kalman filter as well. I think since 2011, I say modern, since 2011, they've tended to offer it. Um, and I think you can potentially ask and pay for them to reprocess older tags
Oh,
to get the Kalman filter data.
oh, great. Uh, we've got a person that's interested in the sim post function, and they're curious if there's a plan for full imputation with simulation from the posterior consideration of the parameter estimation uncertainty.
The simulation section is developing. I think Ian's done some updates since I published the Harpseal paper. And I think there are some potentials for imputation. I'm not I'm not up to date with the latest developments, I'm afraid.
But that might be a good one to go to the GitHub
Yeah,
repository and ask about.
yeah, you could do a
Um,
pull request if you're happy with your coding, and or there's a feature request section on the page as well, so you could certainly post it there.
let's see. Is there any way to make a hierarchical model with aniMotum?
So the joint move persistence model is hierarchical because there's a shared parameter between individuals for move persistence calculation. At the moment, there is no way of fitting a hierarchical state-space model to the movement data itself. Partly for speed, it's done individually.
There's someone who's asking how you would recommend determining movement states for seals. They're studying potential relationships between seal behavior and nearby vessel traffic. And they want to account for the Argos error and the AIS location error. GPS maybe? Hmm.
Um, so the standard thing would be to fit the Argos tracks from the seals to aniMotum, but I could see an appeal to fit the vessel AIS data. It's not something that's been done to my knowledge, but if you have a measure of the location of the vessel and a measure of the uncertainty in the vessel, then those two things should be able to be passed to aniMotum. But I haven't done it and I'm not 100% sure. I'm trying to work out who that person might be, given that I used to work at the Sea Mammal Research Unit and they do a lot of work on seals and vessels. So the other option would be to send me an email.
Great. Any tips for using this on VHF telemetry points with error taken from a GPS?
I mean, it would work. I'm not sure if you've got irregular radio telemetry locations and you're happy with the uncertainty. I mean, I think I would argue that is the uncertainty in the location of the animal from the VHF true to the location uncertainty from the GPS in your hand, if that makes sense. I imagine that the animal might be moving more than that GPS device. Um, but I certainly don't see why you couldn't pass those VHF. Well, essentially, there would be a GPS coordinates. If you've got lats and long coordinates and they're irregular, you could pass them to aniMotum. You'd be kind of doing it custom, as some of these other questions have been. So the question would be how whether or not you can pass the correct uncertainty measure. And then essentially, the way of thinking about it is that each of these GPS points, you're just putting a big circle around it. that's a certain size depending on the uncertainty that you have in that location. And then aniMotum is fitting a correlated random walk through those data. And so you'll get quite close and you'll get closer if you're very certain of that location. So in the case of the PTT and GPS data that someone mentioned earlier, then the state space model is going to try and get quite close to that location because that's more true than the Argos location.
For the whole group. So the person that asked about the seals, they're a PhD student from MSU, and they'll reach out to you. But there's another person in the group who's also interested in this question. So I will be removing the poll everywhere notes after this. So take a look, and if you've got a question about, there's an email in here. If you've got a question about the email, let me know and I can help you out.
Yep.
But yeah, lots of interest, which is great. And there's been a number of questions or follow-up about the error issues that people are having. And again, I think it's if people are having issues, remove the aniMotum and TMB packages and reinstall again, and then reach out at the GitHub repo too. Sounds like that's another way to
I think
get some
if
help.
you it's difficult obviously within this forum to kind of exactly understand that, but I've done a bit of troubleshooting with some of these in the past for kind of one-to-one practicals or class practicals. Because we do face these issues, I would go to the GitHub and just follow the instructions quite closely. It sounds like the install's not worked, in which case you can download the binary versions and that should work.
Great. All right. I think we've covered pretty much all the questions. I know we're getting close to time, and thank you for taking the time and sharing with us. This has been really great, James, and appreciate your help.